So today we can continue with our discussion on bioinformatics. I think somebody else is also joining. Okay. So in continuation to our discussion on the previous class, we will see the major research areas in which these bioinformatics and its tools were applied. These are the list of the major research areas in bioinformatics. Basically, we can see it is used for pattern recognition, then data mining, machine learning algorithms, modeling of evolution, sequence alignment, gene finding, then genome assembly, drug designing, drug discovery, etc. Drug target identification, protein structure alignment, protein structure prediction, then gene prediction, protein-protein interaction studies, comparative genomics, transcriptomics, proteomics, and systems biology, that is modeling of biological systems. So in all these fields, and the diverse fields of biology, we can have applications for such a bioinformatics tools. We will see some of the examples in detail. When we discuss about sequence comparison, if you have similar sequences from multiple sources, for instance, we are taking kinase enzymes, different kinase enzymes from different organisms, and try to compare that sequence, whether it is similar or dissimilar. Then you can use an alignment method so that the sequences were arranged side by side, just as shown in the figure, and see wherever what are the regions of similarity and what are the regions of differences. So that can be compared by aligning these sequences or arranging the sequences side by side. Here, a lot of organisms like elephant, mouse, rabbit, rat, so bovine, all organisms, all the kinases obtained from such organisms were arranged side by side so that they can be easily compared. So such comparison will enable you to understand what are the similarities and differences between these sequences. So this is one of the application of bioinformatics. Then the comparison of these genes within a species or between different species can show similarities between the protein functions or relation between on the species or the compared species. Because if you have a very similar sequence obtained from two different species, and that similarity in the sequence means they are actually intended for producing a protein with similar function. So from the similarity in the sequence, we can derive to a conclusion that the protein products which is produced from that two different species were actually having some similar function. So that prediction is possible by sequence comparison. Also, it will enable you to study the relationship between species. For example, you are comparing 16S rRNA or rDNA sequences for identification of organisms, especially microorganisms, bacteria, and that will enable you to compare the species and the similarity in their sequence. 16S rDNA sequence implies that they were very related species, or they can be arranged in the evolutionary tree in a very close position. So that can be possible, or that is possible only if you compare the sequence and find out whether it is similar or dissimilar. Then the comparison of sequences in order to find similar and dissimilar in compared sequences. Sometimes your intention is just to compare the sequence, whether it is similar or not, or what are the differences in the sequences. That will be useful for the genetic engineering studies and other things. So if you want to, or even protein designing, etc., So you want to compare the sequences and find the similarities or differences in the sequences using sequence alignment. That is one of the purpose. And another one, identification of gene structures, reading frames, distribution of introns and exons, regulatory elements, etc. All these things are possible by comparing your sequences. So that is some of the roles of sequence comparison in bioinformatics. Now, it is also applied in computational evolutionary biology. Trace the evolution of a large number of organisms by measuring changes in their DNA rather than through physical taxonomical analysis or physiological observations. You can depend on such sequence information for very reliable studies on evolution, because that will give you a very clear-cut idea about the relatedness of the species. And previous methods of classification actually that were based on physical analysis of the structures or behaviors or such physiological functions, etc. So such type of taxonomical arrangement is actually it is not that much accurate when you compare with the methods that is depending upon the actual DNA sequence. And then we can build complex computational models for populations to predict the outcome of the system over time. So if you have a better understanding on the sequence and the evolutionary methods, then you can even model computational systems so that the possible evolutionary outcome of a system, of a biological system after a few years or a few decades can be predicted. So what will be the direction of the evolution that can be predicted based on such computational analysis of the sequence. Then prediction of protein structure and function. We can predict the function of a gene if the sequence of gene A, whose function is known, is homologous to the sequence of gene B, whose function is unknown. So you have two sequences and you have the sequence A and sequence B. They are actually homologous sequences with a with a very high sequence similarity. And you know the function of sequence A and you don't know the function of sequence B. But if they are similar sequences or very similar sequence, then you can assume that the sequence B will have or it is sharing the common function as that of sequence A. So that is the concept. So one could infer that B may share the function of A because it is a homologous sequence with a similar sequence. Okay. Then modeler is one of the best softwares in homology modeling. So if you want to predict the structure of a protein from a nascent polypeptide or from the primary structure of the protein, linear amino acid sequence. If you want to predict the structure, three-dimensional structure of a protein, then you can use such tools like the program modeler. It's a software that can be used for homology based modeling of the three-dimensional structure of the protein, and your input will be the linear protein sequence. The secondary structure and tertiary structure, etc., will be modeled by the software. Then protein data bank. That is a database of 3D coordinates or three-dimensional structures of such proteins, and it's a popularly used database. We will discuss it and we will have some practical sessions also on protein data bank. So protein data bank is a database of structures, three-dimensional structures or tertiary structures of the proteins, tertiary and quaternary structures of the proteins. So next one is about gene prediction. Gene prediction is you are actually looking into the genome and look at the genome and predict where genes are present. So you know that there are about 3 billion base pairs in the human genome, spanning the 23 pairs of chromosomes. And to find out the expected 25 to 30,000 genes as predicted, all the genes were not always expressed or coded. And if you want to to locate the start and end of the gene in such a large data set like 3 billion base pairs, where it starts and where it ends, and you want to locate the position of a particular gene in these 3 billion base pairs. You want to find out the position of the gene. So you can look into the entire genome and predict the start point and end point of the gene. And also, as we know, the eukaryotic genes were having introns in between the exons, which needs to be spliced off. So the position of the introns and exons needs to be determined, so that if you are computationally trying to translate a protein without actual transcription, then from the eukaryotic gene needs to be spliced computationally by removing the introns, and that is possible only using optimal bioinformatics tools. So you have to identify the position, start and end of the gene, and also you have to identify the intron regions, so that needs to be removed, and the exons needs to be computationally joined, so that the actual mRNA construct can be created or computationally transcribed. The gene, eukaryotic gene, needs to be computationally transcribed by removing the intron sequences, so that it can be translated again computationally, and the possible protein sequence can be predicted by computational methods. So such type of gene prediction is possible using computational methods. Then another one is structure prediction. Structure prediction is the method of deriving the three-dimensional structure or of the protein from the linear protein sequence or the primary structure. So we can predict the three-dimensional structure of a protein from its primary sequence, and there are various methods, as I mentioned in the previous slide, like softwares like Modeler can be used, which depends on homology modeling, and also there are ab initio prediction methods, and we will discuss it in detail. This computational structure prediction will be discussed in detail in another session. So this is one of the applications, structure prediction from the linear protein sequence. And now you know that you can just from the database sequence of 3 billion base pairs, you can look for any gene, start and end of the gene, you can locate the position of the gene, you can computationally transcribe that into an mRNA sequence, for example, by removing the introns using computational tools, and that can be translated computationally into a protein sequence. So this protein sequence, maybe it is not yet actually translated and or in the cell. This may be a sequence of the protein that is actually computationally translated from the original gene. So a eukaryotic gene needs to be processed computationally by locating the start and end point of the gene. By removing the introns, you need to computationally transcribe that gene into the mRNA sequence, and from that mRNA sequence, again you have to computationally translate into a protein sequence that is a linear protein sequence or primary protein sequence. Again, that can be computationally modeled into a three-dimensional structure. So using such computational tools, a database gene sequence, like the entire DNA sequence of an organism, can be analyzed, and you can locate the genes, predict the genes, and you can even predict the structure of the proteins that is actually that is coded by that is possibly coded by the gene. And another point is on drug designing and phylogenetic analysis. Okay, drug designing is the approach of finding drugs by design based on their biological targets. So actually we are finding or designing new drugs based on their biological targets. And one method is a computer assisted drug design, or CAD, which uses computational chemistry to discover, enhance, or study drugs and related biologically active molecules. So this is a very useful technique nowadays applied, because in the previous days, if you want, for example, now we have the coronavirus, and you know that the vaccine is actually developed in a very short period. Within about 6 months, the development of the vaccine was completed, and provided the sequence of the coronavirus is available and the spike protein, that sequence is available, then you have such methods of designing that structure computationally, and testing maybe thousands or millions of possible drug compounds which is available in the database for that such chemical structures that may be potent drugs that can be tested within a computational system such as in a virtual environment. So the drug receptor and the drug, oh sorry, or the viral protein, maybe that's a receptor, and that receptor and the drug interaction can be tested computationally using such a modeling and simulation tools. So that type of computational analysis of efficiency of the drug is known as a computer assisted drug design. And nowadays most of the pharmaceutical companies were using such methods because it will enable such companies to save the R&D cost. Previously, it may take maybe at least 5 or 10 years to identify the optimal drug compound and for testing and also predicting the efficacy of the drug. So it may take at least 10 to 15 years and to develop a drug. Now it is so rapid that within months such entire testing and identification of the optimal drug compound and its testing and possible outcome predictions and even side effects prediction, everything is done computationally, so that the previous 15 years or 10 to 15 years duration of forest discovery and the entire research cost maybe billions of dollars that is saved by such computer assisted drug design. So this is one of the leading areas in which such computational tools or bioinformatics tools were applied. Okay, now phylogenetics is another area in which we are predicting the genetic or evolutionary relation of a set of organisms. So we can study the relationship of the organism, genetic or evolutionary relationship of the organisms using such sequence comparisons. And maximum parsimony and maximum likelihood are two statistical methods that can be used for analyzing the sequences for studying their relatedness, especially the phylogenetic analysis. So there are two methods like maximum parsimony method and maximum likelihood method, and that is used for constructing the evolutionary tree or phylogenetic tree based on sequence comparison. So there's another phylogenetic analysis. It's a illustration. Now, revolutionizing treatment. As I mentioned earlier, drug discovery, that is a leading area in which new potent target molecules or drug molecules were computationally selected and tested in computer-aided drug discovery methods. Now then, personalized medicine is another concept in which we can correlate between gene expression and a disease. So there are gene chips or microarray like methods in which we can quantify the gene expression in different tissues under different conditions. So you can compare the patient and normal samples or different condition samples. You can analyze all these things, and all the mRNAs expressed in such samples can be entirely isolated, and that can be loaded into a gene chip or a microarray chip, and see the expression of each mRNA or each gene. Each mRNA means the gene is expressed. So the quantity of the gene expression can be measured in terms of the actual quantity of the mRNA that is coded in the system. So for every sample there will be a pool of mRNAs. So the maximally expressed mRNA is actually the most prominent mRNA which is coded in that cell. So that will give you maximum density. The other ones, or if one mRNA or one gene is not expressed, that means mRNA is not produced. So you will not get any color or intensity in the microarray chip. So you can analyze the entire mRNAs in a sample, like a blood sample or something, so that all the possibly expressed genes can be simultaneously screened in a microarray chip or a gene chip, and you can test them in different conditions. For example, a disease means, for example, a patient sample and a normal sample, and the two samples can be analyzed using gene chips and see the difference. And what are the proteins which is highly expressed in the case of the patient, and what are the highly expressed genes in the case of a normal one. So the difference will give you an account of the genes that is active during the disease condition and suppressed during the disease condition. So that comparison is possible by correlating the gene expression of such a patients. And this will help you to enable actually design personalized medicines. As you know, nowadays medicines actually they are becoming more and more personalized. Personalized means the drug that is given to you may not be applicable to the third person, or it may not be useful for me, because I have my own genetic makeup, and the medicine that should be targeted to me should suit my own genetic makeup. That means initially such gene expression pattern in each every individual can be analyzed and see what are the genes which is upregulated and downregulated. And then if you have a better understanding of the upregulation and downregulation of the genes in a patient, then you will have a better understanding, and also you should have a better understanding on the metabolic pathways in which such drugs are interacting. Then based on which you can design a or you can choose a proper drug so that the actual pathway where you want to interfere with the drug can be determined based on the upregulation and downregulation of the genes. So such type of personalized medicine, so that can be decided based on such a gene expression studies. And also there are very different drugs for there are very efficient drugs for HIV. You know that, and normally you know that there will be many viruses in the blood, and you will give a drug, and after a few days you will have a reduction of virus in the blood of the patient. And again you will give the same drug, and after a few days maybe the virus may increase and the patient will dies, because the virus mutates and some of the viruses become resistant to the drug, and they multiply, and there will not be further effect on the drug after the initial stage. Then that affects the patient, then patient dies. So this is what happens due to the drug resistance, and the resistance is actually created by mutation of the concerned pathogen or the virus within the patient, and the drug once effective will fail due to the resistance development by the concerned virus. So one second. Is there any solution? The solution one is you can try a cocktail or a combination of drugs. Usually such nowadays you know that even for bacterial diseases we have a combination drug therapy that is applied by the practitioners, because a single drug, there is every possibility that the bacteria may develop a resistance against the single drug application. So most of the tablets that is nowadays available, or the antibiotics that is nowadays available, may be a combination or a cocktail, so that the antibiotic A and B will be mixed together in a proportion and it will be given, so that the possibility of drug resistance can be minimized by such cocktails or combination of the drugs. So that's one solution. And another one, you should not give the drugs for for the virus which is already resistant. So if a drug is already, or if there is a possibility that the viruses were already resistant to the concerned drug, then you should not give that drug to the concerned patient. But and for example, if one was infected from a person who receives a specific drug, so actually I should have a primary contact, and that person from which I am acquiring that disease might have be treated with some drugs. So there is a possibility that within the previous patient, the concerned virus may have acquired a resistance against that drug which is applied in that patient. So there is every possibility that the virus which affects me or the bacteria that affects me may have already acquired that resistance. So it is better not to give that drug to me or the second person. So it is one of the possibility. Avoid usage of such drugs where such a drug resistance possibility is already there, or if the virus is previously exposed or the bacteria is previously exposed to the concerned drug, then avoid that drug in the subsequent trials. Now, how do we want to know to which drug the virus is already resistant? Because if you want to avoid a drug, you need to certainly know that this is a drug that is already in use. So it is better not to use that in future. So you should have a prior awareness on that one. So one possibility is that you can use sequence comparison from of the HIV, for example, the DNA sequence can be compared, and from patients who are treated with drug A, you will have 3 sets of sequences. And again, you have some patients who are never treated with the drug A. So you have 3 sets or a set of DNA sequences of HIV from different samples. And the first set of population is treated with the concerned drug, and the second set of population is a normal, and that means they are never treated with the concerned drug. So compare the sequences. So after the sequence comparison, you can see some specific differences. For example, in the case of drug A treated cases, we have a mutation here. The sequence alignment and comparison will give you a specific, all the T's actually they were replaced by G in this case. And that can be attributed to the drug resistance. So the drug resistance actually contributed by that mutation. If you identify that sequence, then in further case of HIVs, before treating, you can identify the sequence and look into that location, whether the concerned mutation is there or not. And in that case, it's a very easy example, because a straight mutation can be analyzed, and you can predict that, okay, this is already a drug resistant one and the other one is not. So you can decide based on this analysis. But this is a very easy step. You will never get such a result in actual practice, because mutations are not that simple. It will not always occur in a single position, so that you can easily compare them. So usually the sequence comparison will have multiple locations with different possibilities of changes. For example, here you can see that the A is also there instead of G and T. So a consensus sequences or most predominant changes can be analyzed, but always you can't expect all the strains were having the same mutation. So there is a slight difference. There will be slight differences in the sequences, and that needs to be analyzed and statistically analyzed, so that what are the maximum probable changes there. And you will not get the exact same mutation on all the strains. There will be slight differences, or alternative basis with almost similar functions will be placed at the same location. So it is not that easy. An example, and this will be a classification problem. You cannot differentiate so easily between the drug treated ones and the normals using such a sequence comparison in case of a larger stretch of DNA sample. Now, such a sequence comparisons or larger structures of sequence comparisons can be compared using machine learning tools. In the previous case, actually, you are just reading visually and you can identify the sequence if it's only a simple mutation. But such complex cases in which multiple mutations at multiple locations and the random changes, if you are trying to compare them, you require specific machine learning tools or computational programs based on statistical analysis or statistical methods. You require specific comparison methods. So that means you can use decision trees, linear regression, linear discriminant analysis, and neural network support, then support vector regression. There are various statistical methods. So these methods, or or the machine learning tools or programs or algorithms that is based on the statistical techniques can be applied for such sequence comparisons. And this paper, actually it's in 2006, and that paper describes the prediction possibility of drug resistance. But it's only up to 80% accuracy. It's not 100% because the sequence comparison is not that much clear cut. So it's only a prediction. And prediction with at least 80% accuracy, you can predict that this virus will be or the concerned virus or the sequence that you are analyzing, the virus may be resistant or not. That can be predicted up to 80% accuracy, not up to 100%. So this is one of the possibility of a sequence comparison and its application in case of in finding drug resistance or disease conditions, etc. So this is all about some of the tools or that is used, or some of the applications of the bioinformatics, and we will see that in detail in one of the in the next sessions. Is there any questions?